As many studies show that the antibody level will reduce significantly after 6 months, many people are concerned if the vaccines can protect us for long term. In this video, we will explain how each vaccine induces immunity and why we should not be worried about the dropping antibody level. Upon infection, our immune system will naturally fight against the virus. If we recover from the disease, we will be protected by our activated immune system, so we are less likely to fall sick due to the same virus again. The function of vaccine is to train our immune system without bearing the risk of being infected by the virus. Before we discuss about vaccine, we will examine how our immune system can fight the virus upon infection. Firstly, before the virus enters our cells, our B cells will produce antibody to attack the virus. If this is successful, the virus will be killed before entering our cells. As the virus are removed, our B cells will stop producing antibody, so the antibody level will naturally drop. However, a special type of cells known as memory B cells will be produced. We will talk about the function of memory B cells later. Secondly, if the virus is able to enter our cells, our cytotoxic T cells will be able to recognize and kill the infected cells. In this case, the infected cells and the virus within it will be killed, so the infection will stop. Similar to B cells, after the mission is accomplished, memory T cells will be produced. So, how can vaccines train our body to fight the virus? This picture shows the structure of a COVID-19 virus. The virus contained 4 important proteins. The S protein is also known as spike protein. It is the most important part of the virus as it allows the virus to enter and infect our cells. Pfizer and AstraZeneca vaccines train our body to attack the S protein so the virus will not be able to enter. The M protein allows the virus to have a round shape, while E protein allows the virus to have high infectivity. However, early study in mice showed that immune system does not produce much antibody to attack both M protein and E protein. Hence, no vaccine is produced to specifically target M and E proteins. The remaining N protein is the most abundant viral protein and can trigger the production of high amount of antibody and cytotoxic T cells. However, early study in mice showed that N protein will induce pneumonia as a symptom, so no vaccine is produced to specifically target N protein. So, does it mean that producing antibody and cytotoxic T cells to target proteins M, E and N are useless? Not really, it is just that if we are going to choose only one protein to target, S protein is the best choice. Is there any vaccines that can target all 4 proteins? Yes. Inactivated virus vaccines such as Sinovac can induce antibody that target all 4 proteins. Does it mean Sinovac is better than Pfizer and AstraZeneca that only target S protein? Not really. Each vaccine technologies have their pros and cons. We will explain this soon. This picture shows the component of a Pfizer RNA vaccine. The outer layer is a membrane that will allow the RNA in the middle to go into the cell. A human cell can be divided into 2 parts, the nucleus and the cytoplasm. Our DNA is inside the nucleus. Once the vaccine goes into our cell, the outer layer will be removed, and the RNA will stay in the cytoplasm. Within the cytoplasm, the RNA will direct the cells to produce a lot of spike proteins. These spike proteins will be recognized by B cells and trigger them to produce spike protein-specific antibody and memory B cells. However, this method cannot trigger much cytotoxic T cells response. The advantage of this method is that a high level of antibody can be produced to target the most important protein for the virus, which is the S protein. In addition, RNA vaccine is relatively easy and cheap to manufacture compared to other types of vaccines. However, as RNA is relatively easy to be destroyed by our cells, the amount of time that our immune system is exposed to S protein is quite short. Hence, booster dose is likely to be needed. Besides, this is the first time that RNA vaccine is used on human. We are not sure if there will be any long-term adverse effect. However, theoretically RNA vaccine should be very safe and will not have long-term adverse effects. As RNA vaccine usually requires ultra-low temperature during transportation and storage, it is not suitable for areas that are difficult to reach. Finally, as this method only targets the spike protein, if the virus mutates its spike protein, the effectiveness of the vaccine will greatly reduce. This picture shows the component of an AstraZeneca virus vector vaccine. The outer layer is a virus capsid, coded by E1 and E3 genes derived from adenovirus. The vectored DNA in the core contain the genetic codes for spike protein, E1 gene and E3 genes. The capsid allows the vectored DNA to enter the nucleus. Inside the nucleus, the vectored DNA will produce RNA. The RNA will produce spike protein in new virus vector vaccine. The spike protein will be able to trigger antibody production and cytotoxic T cell response. The new vaccine vector will be able to enter other cells and trigger more immune response. The advantage of this method is that a high level of antibody can be produced to target the most important protein for the virus, which is the S protein. As the immune response includes both B cells and cytotoxic T cells, the protection will be even stronger than Pfizer RNA vaccine. However, the cost and process to produce this vaccine is higher and more complicated. As this type of vaccine has only been used in 2019 for Ebola, we also do not know if there is any long-term adverse effect. Because this method only targets the S protein, if the spike protein gene is mutated, the efficacy of the vaccine will be reduced. Finally, it has also been reported that this vaccine is much more likely to induce venous thromboembolism as an adverse effects compared to other types of vaccine. This picture shows the components of inactivated virus vaccine like Sinovac. Basically, it is the whole COVID-19 virus which have been killed with methods such as heat or formaldehyde. As the vaccine contains the whole virus, it will be able to trigger antibodies that target all the components of the virus, including S, M, E and N proteins. Hence, if the virus mutates the spike protein, the antibodies targeting other proteins may still provide some protection. Besides, as this type of vaccine has been used since year 1896 for typhoid, the long-term adverse effects of the technology is very well documented. For immune-compromised people, or people that are likely to get adverse effects from other types of vaccine, inactivated virus vaccine is most suitable as it has the least amount of adverse effects. However, this type of vaccine is relatively expensive to produce even though the procedure is quite simple. Another disadvantage is that, due to the low amount of spike protein contained in the inactivated virus, strong immune response cannot be induced. The antibody level will be low compared to other vaccines and cytotoxic T cell response is not available. Now, we are clear about how each vaccine triggers immune response and the pros and cons of each technology. But as we mentioned at the beginning of the video, the antibody level will drop significantly after 6 months. Are we still protected in long term? For virus-vectored vaccines such as AstraZeneca, as the vaccine triggers both antibody and cytotoxic T cells, the cytotoxic T cells can protect us even though the antibody level is low. Hence, we actually observe much stronger and consistent protection from AstraZeneca than other vaccines. You can find this comparison from our previous video. For Pfizer and Sinovac, as both vaccines only trigger antibody production but not cytotoxic T cells, the protection will drop if the antibody level reduces below threshold. However, as we have the memory B cells triggered by vaccination, we can quickly reproduce the antibody if we were infected. This is called secondary immune response. The time needed for the secondary immune response to produce sufficient antibody is roughly 5 days, but the virus only needs about 3 days to induce mild symptoms. Hence, the memory B cells will not be able to prevent us from getting mild symptoms, but it will be able to prevent us from developing severe symptoms that require hospitalization. In conclusion, each vaccine has its pros and cons. We should take any vaccines available to us, and take booster in the future to further strengthen our immunity. It is natural that the antibody level will drop over time. It is nothing new that we have never encountered before. However, our memory B cells will be able to quickly produce antibody again to protect us from developing severe symptoms. Since the COVID-19 outbreak, a lot of healthcare resources have been focused on dealing with the pandemic. This leads to the lack of attention of other important matters such as cancer, diabetes, obesity, mental health, etc. As we are going into the endemic phase, we will start to move on to other health topic. In our next video, we will discuss the most common types of cancer in Malaysian, with detailed breakdown according to gender, race and age. If you enjoy the information that we share, please like this video and subscribe to our channel so that you will be notified when our next video is ready. By giving us positive feedback, you will help to promote our videos so that more people will be able to receive correct information regarding COVID-19 and other health analytics. If you are interested in vaccine and infectious diseases, you might be interested to join this short course in immunology and infection. You can complete the course in 3 to 6 months and the course is conducted fully online. For more information, Please scan the QR code or refer to the link in description. If you prefer a full master degree instead of short course, you can join this program. You can complete the whole master program in 1 to 2 years via online or mixed modes. For more information, please scan the QR code or refer to the link in description.